Welcome, everybody. My name is Terri Conneran, and I am the founder of KRAS Kickers. And mostly it's because I'm a lung cancer patient. And I got to tell you, I am beyond thrilled this evening that I'm able to bring as a guest speaker to you, Dr. Mark Awad. And let me pull up my little notes here so I don't blow anything here. Okay. He is a clinical director of Thoracic Oncology Treatment Center, assistant professor of medicine at Harvard Medical School, at thoracic oncology at Dana-Farber. And I'm sure above and beyond that, you've got many other titles, of which you're just too humble to share with us. But I am just totally thrilled that you're here to join with us tonight and talk to the KRAS Kicker community about kicking cancer's KRAS. So welcome.
Thank you so much, Terry. Thank you for everyone for dialing in. It's a pleasure to be here. I wanted to send a warm greeting from sunny Boston, where we're enjoying a beautiful spring day today. Yeah, Terry, as Terry said, I'm a thoracic medical oncologist at the Dana-Farber Cancer Institute here in Boston. And I finished my fellowship training here in the Dana-Farber program about 7 years ago now. So I've been on the faculty for the last 7 years. Oh, okay. I see patients in clinic, in our thoracic clinic, with lung cancer, and I also spend a fair amount of time doing what we call translational research, or trying to understand why certain therapies work especially well in some patients and not as well in others, or trying to also understand how some cancers can evolve to develop resistance to treatment so that we can then try to identify and develop even better therapies to fight cancer. So it's a pleasure to be with you all. Thank you for taking time out of your day to join in, and I'm looking forward to a great discussion. I see a few familiar and friendly faces in the group already. So.
Well, thank you. Well, we're excited to hear what you have to say. And so, just kind of like to kick things off, would you give us kind of like your explanation is what is a KRAS lung cancer?
Right. So, over the last almost 2 decades now, we've been doing a lot of DNA analysis of many cancers, including lung cancer. And this is so important because we have some sense of risk factors for cancer in general, but we don't always know what causes a cancer. We don't really, in some cases, have a sense of why the cancer formed or how long it's been there. But by looking at the DNA of lung cancer and many other cancers, in many cases, we can find that what used to be a normal gene in the DNA mutated or changed the DNA sequence. And when that happens, it kind of turns on the accelerator, turns on the gas on what used to be a normal lung cell that shouldn't be really growing or dividing too much. But when a mutation occurs and creates this signal to tell the cell to start growing and dividing, then we can identify in some cases why a cancer has been developing. And when these genes are mutated, we call them oncogenes or driver genes because it kind of drives the cancer's growth forward. Those cells start dividing, multiplying, growing to form a tumor. And that cancer can also potentially spread or metastasize to other parts of the body. So
right here.
Sorry, I think I just heard a little bit of talking in the background. If people maybe can stay on mute if you don't mind, thank you, so we can all hear together. So we know KRAS is actually one of the most commonly mutated genes in cancer in general, not just lung cancer, but especially in lung cancer, it is very frequently mutated or abnormal. And when we see that, that explains what's really driving the cancer's growth forward. So there are different specific subtypes of KRAS mutations depending So KRAS is a protein, and a protein is made of a string of amino acids, all kind of like a string of pearls, where each pearl is an amino acid, and they all kind of string together to form this KRAS protein. And so there can be mutations at different positions along this string, and we number the pearls on the string essentially, so that we can sort of tell where along the protein the mutation or the changes occur. So you might see some of these notations if you see access to your notes or your genomic DNA test results. You might see something that says KRAS G12V or G12C or G13 or Q61. So what does that mean? The number represents the position along the protein that the DNA change occurred. And then the first letter is the normal amino acid that should be there at that position. And then the letter that comes after the number is the change or the mutated amino acid. So for example, this notation G12C means that at position 12 in the KRAS protein, what should be a G for the glycine amino acid changed to a C, which is cysteine. Or instead, if it's G12V, that means a glycine changes to a valine, et cetera. So these are the mutations that we sequence on the DNA to try to understand how the cancer developed, but also importantly, can we take advantage of that to develop therapies that are specific to that mutation?
Okay, well, that sounds good. Okay, so let me see if I repeat it back to you if I picked up right. Okay, so like a string of pearls, each spot has a number, and on the number, if it's supposed to be, it should be G12, and if it was fine, it would just be G12. But if it was G12 A or V or C or D or whatever, that means it was substituted with the letter instead of the G, correct?
Exactly right. Yes.
Okay. Well, then you did a great job explaining it.
Okay. That's great.
Okay. Do we have any idea why it's KRAS that keeps causing the cancers?
You know, it's such a fundamental protein. So, you know, cells at one's, at everyone's point in life, need to grow, need to divide, especially, during embryo fetal development and after, baby is born, the baby, of course, has to grow and get bigger and bigger. So there are normal pathways that we call them to instruct cells when to normally grow and divide during growth and development. And so KRAS is really at the crux of these normal signaling pathways. Normally, a cell needs a signal to tell it to grow and divide. And some of those signals we refer to as a growth factor. The growth factor in the bloodstream or around the cells binds to something on the cell surface that instructs the cell, okay, now it's time to grow. So the cell should divide and multiply. And right at the inner part of the cell, right after that growth factor binds to its receptor, is KRAS. And so it's right at the sort of center point of this major normal pathway that, when needed, instructs the cell to start growing, dividing, multiplying. And normally, after growth,
That's why she brought two.
Y'all could please mute yourself. That would be great. Thank you.
Yeah, normally once growth sort of plateaus, you shouldn't see this signaling going on and on. But when a mutation occurs, it kind of tricks the cell into thinking, okay, now it's time to grow again. And so that's where KRAS is a important and normal protein in normal growth and development. And when mutated, it kind of hijacks these pathways in the cell to tell the cell to start growing and dividing even without any signal to do so.
So it's at a crucial intersection of where normal growth happens.
Exactly right. And that's not specific to lungs. And so KRAS can be mutated in other types of cancer like colon cancer, pancreatic cancer, and several others. That's kind of tells you it's at such a kind of a key focal point in normal cell processes. Yeah.
I read somewhere, and please correct me if this is wrong. I read somewhere about 20% of all cancers they believe are KRAS mutant cancer.
Yes, it's probably one of the most common what we call oncogenes, meaning a gene that when mutated causes cancer. That's right.
So we're in good company here.
Right, it's very common, and we've known about KRAS mutations for decades actually, but until more recently, it's been harder to target very specifically. So it's a common gene mutation across many cancer types.
Right. Well, that's great. Now, what is it that you see? I mean, because I mean, it's not all about the problem. So we've identified the problem. What kind of solutions do we have coming?
Right. Well, we've made some great strides in the treatment of lung cancer in the last several years. Some people may have heard of other targeted therapies for other genetic lung cancer subtypes. So when I meet a new patient, we do DNA testing, and usually we find, in many cases, one gene is mutated that has explained why the cancer is growing. So KRAS is one of them, and then there are others like EGFR, ALK, ROS1, and others. And so in these other cancer types, we have FDA-approved targeted therapy pills that are very effective at treating EGFR mutant lung cancer or ALK positive lung cancer, et cetera. So that's been one major development for lung cancer, is the development of targeted therapies. And now we're just seeing a wave of targeted therapy, exciting developments for KRAS. The other major advances that we've made are not only improvements in our chemotherapy and our supportive care to make chemotherapy more easy to tolerate, but also the advent of immunotherapy or immune checkpoint inhibitors. These are drugs many people are familiar with. They make see commercials on TV. Drugs like nivolumab, which is Opdivo, or pembrolizumab, which is Keytruda, and others. And so we know that KRAS mutant lung cancers, a subset of them, can be very sensitive to immunotherapy. These are drugs, infusions that stimulate the body's immune system. We know the immune system in general is trained to recognize the difference between what we call self and non-self. So your immune system shouldn't attack yourself. It should only attack foreign invaders like viruses, bacteria, et cetera. Cancer is tricky because it arises from normal human cells, what used to be a normal lung cell, and it mutates. And so cancer, in some sense, looks like a normal part of the body, but is also a little bit different. And so the immune system at times can recognize that a cancer is a little bit foreign, like it doesn't quite look like the normal body, but the cancer can kind of hide from or evade from the immune system by making these shield proteins or these masks, for example, called PD-L1. And so now we have got these immunotherapy drugs that stimulate the body's immune system. And in many cases, it kind of takes those brakes off the immune system, getting the immune system to attack these mutated cancers. And so some KRAS mutant lung cancers are very sensitive to these immunotherapies, and we see these amazing and dramatic, beautiful long-term responses. Other times, KRAS mutant lung cancers don't respond as well to immunotherapy and are more resistant to it up front. And so we're trying to understand, and we have a better sense of why that may be occurring now in some cancers. But I think these are some of the major inroads that we've made in general in lung cancer. So the development of immunotherapy and the development of targeted therapy pills that are specific to gene mutations.
Okay. Well, you're doing great. I'm gonna have to let you catch your breath. I
Yeah,
don't know
right.
where you are. We gotta brace ourselves. Okay. As far as like we've all heard about the G12C, And there's so many other different types, right? There's some pan KRAS treatments that are coming down the pike that look like they have possibilities. If you'd like to maybe address some of that, and kind of get into just some of the different targeted therapies that we've got coming.
Right. So, I think many people have heard of these G12C inhibitors, and those are a little bit further along than where we may have in terms of being effective at treating this subset of KRAS. And the reason why these drugs are a little bit further along is because that specific mutation that causes that C, the cysteine substitution, you can develop a drug that directly binds to that cysteine, and it's kind of very specific to that C. And because the other mutations aren't quite as amenable to the drug directly binding, so that's why the G12C inhibitors have been a little bit further along in their clinical development. But there are exciting avenues to also try to target proteins that cooperate with or bind to KRAS that it requires for its functioning. So, for example, one is a companion protein is called SHP2, SHP2. There's also some downstream proteins. For example, MEK is another protein that we're trying to sort of perturb. That's MEK. But yes, there are some different strategies to try to develop drugs that will degrade or deplete KRAS from within the cell. So I think there are quite a number of strategies being employed to try to combat these KRAS mutant cancers. I'm seeing also a question through the chat, so I'll try to kind of keep my eye on that too. And Terri, Dusty, feel free
I'll just shout it out for you. Bobby asked, am I correct in thinking that a low PD-L1 is more resistant to immunotherapy?
Right, it's a great question. So PD-L1 is that shield protein that some cancers make to hide from the immune system. And we give it a score from 0 to 100%. And what does that score mean is that if you have a biopsy of a lung cancer and the pathologist looks at it under the microscope, they can do a stain for PD-L1 to try to tell which cancer cells are making this protein and which ones aren't. And then they score that as a percentage. So what percent of tumor cells on any given biopsy are making the PD-L1 protein, 0% all the way up to 100%. We do think it's not a perfect, what we call biomarker. A biomarker is something we can test for on the cancer that helps us understand what a cancer may be more or less sensitive to in terms of a therapy. But it's not a perfect biomarker, but we do think that the higher the level, as it gets closer on the side of 100%, about 50%, 75, 90, 100, on that scale, we do think that the immunotherapy drugs are more likely to work on their own. And if the PD-L1 level is on the lower side, like 1% or 0%, immunotherapy is less likely to work by itself. And that's a setting where we may combine it with chemotherapy so that the two approaches can work together. And so that PD-L1 level is really the target of drugs like Keytruda and Opdivo or pembrolizumab and nivolumab and others. And so if the cancer is not making that protein, that means it's not really using that pathway to grow or to hide from the immune system. And therefore, these drugs which work on the pathway or they perturb this pathway are not as effective when there's not the protein to kind of go after. So yes, again, it's not perfect. So there are some cancers that have very high PD-L1 levels of, say, 95%. That don't necessarily respond to immunotherapy. And then there are some cancers that have very low PD-L1 level, like 1% or so, but they do respond. So We're also trying to understand why that is and develop more precise biomarkers of immunotherapy response. So, why do some cancers respond really well and others are more resistant? PD-L1 is one marker.
Okay, so right now, let me just repeat back, make sure I get it, okay? So the higher the PD-L1, the more likely that you are to respond to immunotherapy. Is that right? Okay. Now I also know I don't see that there's consistency. Like, we've had the same tumor testing and you don't always get the same percent back. Is that a regular incident?
That's a great question, and one of our areas of research that I just had a meeting earlier today at Dana-Farber. So we're looking at this question. Certain things within a cancer we think are fixed, meaning they'll always be present within any biopsy site, and the mutations like KRAS, that tends to be very constant and consistent, regardless of what you biopsy or whether you biopsy something a year ago versus today. You'll tend to see that same marker present throughout the cancer. PD-L1 may be a little bit different, and there might be more heterogeneity in the protein expression. And that's something we are looking at among patients who have had, just as you said, Terry, more than one biopsy, either at the same time, different sites of cancer biopsy concurrently, or different biopsies across time. And we think this PD-L1 level might be a bit more of a dynamic marker that can, sometimes you have higher levels, sometimes lower levels, and we're trying to understand what accounts for that.
Does it depend on where it is, like within the tumor, or just like in the cycle of the tumor or anything, or we just don't know?
We're trying to figure that out, actually. What we know, for example, is early stage lung cancers. When I say stage, like that's stage 1, 2, 3, 4, where stage 1 is really the earliest stage, a small tumor just in the lung. A stage 4 means the cancer has metastasized or spread to other parts of the body. We know that in general, stage 4 lung cancers have higher PD-L1 levels than stage 1 lung cancers. And we think that's because in order for the cancer to have grown and spread, it has to have kind of figured out a way around the immune system. And we think that along the evolution as a cancer grows and spreads, that if it makes this PD-L1 protein, it can kind of bypass the immune surveillance and get to other parts of the body. So we're really trying to understand what are the factors that govern changes in PD-L1 expression.
Yeah. Does it, I completely lost what I was going to say. Does it show up in a liquid biopsy?
Right, good question. So liquid biopsy, so there are two main ways that we look for gene mutations through sequencing of the DNA. One is through a tissue biopsy, so a needle going straight into the tumor and getting a piece out. And from there, once we have a piece of tumor, the pathologist can extract the DNA and send it for sequencing to detect the KRAS mutation. That's really a very sensitive way to look for these gene mutations like KRAS. Some cancers will shed or release DNA from within the cell into the bloodstream or into the circulation. And we call that circulating tumor DNA, sometimes abbreviated ctDNA for short. So circulating because it's circulating in the bloodstream, tumor DNA. And in some cases, by doing a blood test, we can detect these gene mutations like KRAS or EGFR or others. Not all cancers shed that much DNA into the bloodstream, so the blood testing is not always as sensitive as going straight in with a needle biopsy to getting cancer cells to do the testing that way. So there are some advantages and disadvantages of tissue testing versus blood testing. We call the blood testing or the circulating tumor DNA testing, we call that liquid biopsies because you're getting blood instead of a chunk of tissue. So that's sometimes called liquid biopsy. blood biopsy, plasma biopsy, that all, or ctDNA circulating, that all kind of means the same thing. And there are a number of companies that offer this kind of testing, like Garden 360 and Foundation One and all of these other ones. And so yes, KRAS can be detected by liquid biopsy, but not all of the time. It kind of depends on, in some cases, if there's a relatively small amount of cancer or a small volume of cancer, it doesn't tend to release as much DNA in comparison to cancers that are a little bit larger or in a few different places in the body. So a positive liquid biopsy, meaning that you find a mutation in the blood, that's helpful. You can really rely on that to be confident that that's what is present within the cancer. But a negative liquid biopsy test, meaning you send the blood and you don't get any results back, that doesn't necessarily mean there's no mutation in the cancer. It just may reflect that the cancer is not releasing enough DNA into the bloodstream for you to be able to detect.
While they're going,
And Terry, sorry, just to finish, the PDL1 is not a gene mutation. It's a protein level that we measure directly on tumor cells. So the PDL1 level cannot be, thus far, checked on a liquid biopsy. That is one where you have to do it on a tumor biopsy. So I just wanted.
Okay. No, that's a good point. I didn't know that. Now, I know that is for lung specifically, is there any sort of like a screening or like a ctDNA, or like I know that they can do like CTC for like colorectal. Is there anything like that they can do while you're in treatment for lung to see if you're responding to it?
Right, that's a great question. So how do we monitor the efficacy of a treatment? How do we know whether a treatment's working? And the sort of best established standard is with scans, doing CT scans or PET scans or MRIs at regular intervals, where you can measure the cancer at baseline, start a new treatment, and then obtain scans after, say 2 to 3 months and compare. Did the cancer shrink? Did it grow? Did it stay the same? Did a new spot appear? And that's been the kind of best established standard for determining how well a treatment is working. That is typically still what gets reported in the vast majority of our clinical trials, is what's called the response rate, meaning what % of patients who go on to any given therapy respond to the treatment, meaning a response meaning the cancer shrinks down dramatically. But we've published on this recently in our group at Dana-Farber and other groups as well, that circulating tumor DNA or ctDNA can also track with the disease burden or the volume of cancer within the body, such that, and it can be a very early change. So for example, if I get a blood draw on a patient and we see some baseline level of ctDNA, what we did in our analysis that's been published recently is we got another blood draw as soon as 3 weeks later. And that's usually too soon to get a scan. But in many cases, if we saw a very dramatic drop in the level of circulating tumor DNA by that first follow-up blood draw 3 weeks later, that very often could tell you that you would likely see a positive improvement on the next scan. So it was kind of an early window into what the future scan might look like. And conversely, if you see circulating tumor DNA levels increasing early on, that might be an indication that the cancer may be growing. So it's something that we are doing research on to say, how can we use this as another sort of marker to anticipate, is a cancer going to grow or shrink, and get an earlier window into that, such that we can sort of stay attuned to understanding if a therapy is not going to be effective for a patient. We want to know that early so that we can make adjustments or make changes. Or if it looks like it's going to be effective, that can offer us a bit of reassurance as well. but it's not, I would say, a perfect marker. It's not really an FDA approved sort of strategy for monitoring whether cancer is growing or shrinking, but it's, I would say, more on the investigational side. But we hope that as this technology becomes faster and cheaper and more available, that we'll be able to monitor cancers with blood draws serially. Similar to, if patients are coming in for an infusion every 3 weeks, we always get a blood count and some chemistries. These are standard blood tests, and the hope someday is that along with, your white blood cell count and platelet count and all of those things, you'll also sort of be able to get a real-time assessment of the tumor DNA level.
That'll be great. I hope so. I hope so. And I saw one of the questions that was posted in the chat about patients who did not respond to checkpoint inhibitors first time during as first line. Could they try again?
Right. So that's a good question. We, again, are trying to understand these biomarkers of response and also resistance to immunotherapy. We talked about PD-L1, how that's one factor that may influence the likelihood of response to immunotherapy, like checkpoint inhibitors. There are other biomarkers that may predict response or resistance to immunotherapy. So tumor mutational burden is one, which we're happy to talk about. There are also concurrent gene mutations sometimes present along with KRAS. So in addition to the KRAS mutation, sometimes we can see other genes mutated, and sometimes these other genes might influence the likelihood of response to immunotherapy. So we have a research publication about to come out on looking at STK11 and KEAP1, KEAP1 genes and others. And when these genes are co-mutated along with KRAS, these cancers tend not to respond as well to immunotherapy as compared to KRAS mutant lung cancers without those gene mutations. So these are a number of the factors that we're looking for to try to understand why some cancers do not respond to first-line checkpoint inhibitors. We don't have really any substantial evidence yet to date that if a cancer doesn't respond to immunotherapy initially, that it can subsequently respond at some later date. I think it is a interesting idea and concept and thought. It has not yet been borne out or well established in any large, kind of cohorts of patients to get a sense of how well is what's the likelihood of responding to a checkpoint inhibitor down the road if the cancer didn't respond to it initially. What we do know is there have been trials of a PD-L1 inhibitor and a second immunotherapy called a CTLA4 inhibitor in patients who have already been on a checkpoint inhibitor before. So, for example, there was a trial combining durvalumab, which is a PD-L1 immunotherapy, with tremelimumab, which is what's called a CTLA4 inhibitor, in patients who had already been on a prior immunotherapy. But unfortunately, in that trial, there was only a very, very, very small percent of patients that seemed to derive any benefit, probably under 5%. And so we do not tend to recommend going back to an immunotherapy or going to a different drug in the same class. A lot of these companies have the same type of medicine, like there's pembrolizumab, nivolumab, atezolizumab, durvalumab. They're all really in the same class. It's kind of Coke and Pepsi, or how there's different cholesterol drugs that are made by different companies. So if a cancer has grown through one of them, we don't really advise switching to a different one because we think that that might cost some time where the cancer may continue to grow, and we'd rather switch to a clinical trial or another type of therapy that has a better track record or is more established.
Okay, that sounds logical. And like I kind of told you before, as we're kind of getting into the clinical trials and stuff, you did some research on resistance to G12C inhibitors. Can you water it down and explain it to us?
Right. So, for many years, when I was a fellow and as a faculty member at Dana-Farber, one of the areas I've been focusing my research on is trying to not only understand what might predict whether a cancer is initially sensitive or resistant to a treatment, but also what we call acquired resistance, meaning a cancer really responds well, it shrinks down, and it can stay down for long periods of time. But we know in many cancer types, so not just lung cancer, but colon cancer and others, that cancers that initially respond to a treatment and shrink down can develop drug resistance, where then it starts to grow again, or we see a new spot of cancer appear. And that's something that we're really trying to understand, and by understanding it, it really helps us know the biology as to what's going on within the cancer, with the ultimate goal of developing either new treatments up front to delay or try to prevent resistance from emerging, or new therapies to use at that time to try to overcome resistance and get the cancer to shrink back down again. And so we have had targeted therapy approvals for other types of cancer with EGFR mutations, ALK mutations, ROS1. And by studying how cancers have developed resistance to those drugs, that has really fueled drug development to help us develop new therapies to overcome resistance. And those new therapies have now become the standard of care for those types of cancers. So, I know it can be difficult to talk about and hear about drug resistance when, people may be on these therapies or have received these therapies. But I think by trying to really stay ahead of it, and these drugs aren't yet FDA approved, but we're already really trying to understand from patients who so graciously and generously participate in clinical trials. We're trying to develop better drugs, but also understand if there's a point in time when these drugs stop working as well. We need to understand what's going on, why is that happening, so that we can stay on top and develop the next wave of therapies. And so we had presented our initial look at resistance to KRAS G12C inhibitors. One drug is called adagrasib from the company Mirati, or MRTX849. We've also been looking at sotorasib, or AMG 510 from Amgen. And what we did in our collaboration with many other academic cancer centers is for patients who benefited from these drugs in the past, but then where we saw the cancer start to grow, we would get a repeat biopsy, either of the tissue or a liquid biopsy of the blood, and we would repeat the DNA sequencing, and we would compare it to the sequencing from before the patient was treated with the KRAS G12C inhibitor. And we would compare the DNA before versus after treatment and ask what changed between the two time points, and in order to try to understand how the cancer developed resistance. And so we involved patients with both KRAS G12C mutant lung cancer, which was the majority, but we also had patients with KRAS G12C mutated colon cancer, one case of cancer of the appendix also. And what we found was that there were a few different ways the DNA could change that explained why the cancer was starting to regrow. Number one is we saw that new mutations in KRAS were appearing. So besides the G12C, KRAS G12C mutation, we would sometimes start to see the emergence of other changes in KRAS. For example, one is we saw G12V or G12D or G13 mutations, and that explained that there were mutations, other mutations in KRAS that could kind of get around the drug. We also found some new mutations that hadn't been described before in KRAS at position R68 or H95 or Y96. And this is important to understand and recognize because these are brand new novel mutations in the KRAS gene that we hadn't ever really seen before. And in fact, we had to kind of go back to some of the diagnostic companies that were doing these sequencing tests and say, you found this variant, which is reported as a VUS variant of uncertain significance, but it's really significant because these mutations at other positions in KRAS are exactly where drugs like adagrasib and sotorasib bind in the KRAS drug binding pocket. And when you mutate those binding sites, the drug just can't bind to its target anymore. And that explains why in some other cases, the cancer became resistant. So we saw these new changes within the KRAS gene. We also saw changes in other genes that kind of bypass around KRAS. So we saw mutations in a gene called MEK or MAP2K1. That is a protein which we say is just downstream of KRAS. So KRAS has to signal through other proteins. And so by seeing mutations in this other protein, that kind of bypasses around KRAS and propagates this growth signal further. We saw mutations in other genes like RET or BRAF or ALK and others. So in some cases, the KRAS protein mutated further. In other cases, the other genes mutated to kind of work their way around KRAS. In a couple cases, we saw that the type of cancer shifted a bit from, in two cases where they had a tissue biopsy, we saw a change from lung adenocarcinoma, which is the type of non-small cell lung cancer where these KRAS mutations are more likely to be found, to lung squamous cell carcinoma. So the shape of the cancer cells changed under the microscope. And we've seen that can cause drug resistance, for example, in EGFR mutated lung cancer. So in general, we found that the KRAS gene can change again and explain why the drug doesn't work as well anymore. Other genes can change, or the histology, the appearance of the cancer under the microscope can change. And in doing so, we're hoping that will inform kind of a rational strategy. And it may not be a one-size-fits-all strategy for all patients, but if we can understand what's happening in each individual person as resistance may be developing, we can potentially combine novel therapies together to overcome resistance. We tested in the lab, so not in patients, but in the lab, we were able to develop models of these, what we call resistance mutations. Because I see a question in the chat, can we switch from one KRAS G12C inhibitor to another? And we have learned through our study that certain mutations will likely confer resistance to not just the drug that a person is taking, but similar drugs in class. So as an example, this Y96C mutation we found looks like it would cause resistance both to adagrasib and sotorasib. By contrast, a different mutation, H95, might have a little bit of a differential sensitivity or resistance from one drug to another. But you have to also look in the context of any other findings that may be causing resistance. So we don't have any examples of a patient that has been published in the literature yet switching from one, becoming resistant to one drug, switching to another and seeing the cancer shrink down again. But that will be a really important thing for us to test and to watch for. In part, we haven't been able to do that because none of these drugs yet is FDA approved. And they're only available through clinical trials thus far. And most of the clinical trials to date have only allowed patients to enroll if they had not received any prior KRAS G12C inhibitors. So therefore, they excluded patients who were on one drug and we couldn't switch them to another drug. But now, especially if these drugs get FDA approved, we will be able to use them a bit more liberally. And in fact, there are these expanded access programs that are open. For example, the Amgen drug AMG 510 or sotorasib has an FDA breakthrough designation, but there's also what's called an expanded access program or EAP that is trying to make the drug available to more individuals. And that trial does not exclude prior patients who have been on prior G12C inhibitors. So there may be settings where we may try to switch from one inhibitor to another to see if they can overcome resistance. So it's a good question. And I think that may be a strategy that we try in cases that become resistant just through mutating KRAS. So for example, if KRAS has mutated and one drug can't bind, but another drug can still come in and bind, that might be a good strategy. If there's a situation where the KRAS gene hasn't changed further, but another gene becomes abnormal, say BRAF or MEK, that is bypassing around KRAS and causing the signal to kind of still tell the cell to grow and divide. So there, it may not be as good of a strategy to switch out different KRAS inhibitors because that's not really where the problem is. It's these other genes are kind of taking over the signaling. So this will be important to study and to test.
Oh, it's definitely important to have a strategy because we know cancer is smart. That's how we got it to begin with. And so it's going to come up with resistance. And whether it's on the C or one of the other ones later on, we need to be prepared for it. So as disappointing as it initially sounded, it's actually good that we're strategizing and looking at it now.
That's right. And we're, our analysis, was only enrolled patients who developed drug resistance. We also know that there are many patients that have really prolonged responses to targeted therapies as well. And so we're trying to understand why that is too. So we kind of looked at a subset of patients who are receiving these therapies, only that kind of portion who were developing resistance. So I do think I can understand how it might be a bit disappointing or dismaying to initially hear about cancer developing resistance. But I think we are really trying to understand it and stay on top of it in order to develop better therapy.
Okay. All right. So I'm going to hit you with a couple of the other ones specifically because I get these questions a lot. G12A, G12D. We've seen a lot around G12V lately. So if you hit up those two and maybe talk a little bit about a vaccine that we talked about.
Yeah, I think this is such an important field for all of us to be working on because these KRAS mutations are so common in a variety of cancer types. And so, but as we said, there are these different flavors of KRAS mutations. So, right, although we've been just talking for the last few minutes on G12C inhibitors, what about these other types of KRAS mutant lung cancer for which drugs like adagrasib and sotorasib were not designed to target? So there are a number of other strategies to try to develop therapies that will target these other types of KRAS, G12A, G12V, G12D, including using immunotherapies, including vaccine therapies. So what is a vaccine for cancer? I think we all have heard about vaccines plenty this year, fortunately, which is good for COVID and other things. So in general, a vaccine is a small portion of a larger protein that in and of itself is not, doesn't cause problems. It's a small piece of a larger protein that is intended to kind of train your immune system to really ramp up a response against this small piece. In the case of cancer, like a KRAS G12A mutation occurs on a small portion of the larger KRAS protein. And so if we can develop a vaccine using a small fragment of KRAS that includes that A G12A mutation or D or V or what have you, can we create a vaccine from that? And by injecting this small fragment, can we train the immune system to recognize that G12A mutation from the vaccine such that your immune cells, which continue to circulate throughout the body, anytime they encounter another G12A mutation, will the immune cells attack, be trained to attack that. And so that's where these cancer vaccines are trying to come in and say, can we use small pieces of these KRAS proteins or these KRAS mutations to train the immune system to develop an immunologic response against that mutation, and then kind of seek and destroy anytime they find a mutation, seek out and try to kill those cells. So there are some companies out there like Gritstone and Alicio and others that are trying to develop these KRAS vaccines.
How far down the line are they?
They're currently some of them open and enrolling. Some of them are enrolling what is called as a by HLA restriction. So how does your body share or tell your immune system what's going on? If it's a normal cell or a mutated cell, is that inside each cell, the cell chops up proteins into small fragments, which we call peptides, and the cells present peptide on the surface. The way that these little protein pieces appear on the surface are what are called the MHC molecules. It kind of displays pieces of protein on the cell surface. And everybody's MHC molecules are a little bit different, but that's how your cells share information about themselves to your immune system.
So would it be fair for me to say that this MHC is like the skin to the cell? Like
Yes.
It's an oversimplification, but it would be like a skin to the cell?
I would say yes.
I mean, I know you've got to reach scientifically.
No, that's good. That's right. So, and it tells your immune system looks at the skin and it says, is this my skin or is this something else? So cancer, which has mutations within the cell can send these mutations or display them on the skin. But again, it can sort of trick your immune system not to recognize it as being abnormal. And so everybody's HLA type is a little bit different. You can think of it as a blood type, but for your skin, for your tissue. You might, if anyone has had, been involved in a organ transplant or heard about organ transplants, you have to match. You hear about, am I a match or not, or a blood transplant. And when we talk about match, this is what we're talking about, this MHC match. You have to kind of match the skin of different organs so that you don't transplant an organ and the body would reject it. And so, so for some of these vaccine trials, they're selecting certain HLA, kind of certain blood types, but certain tissue types for one type of vaccine or another. So some of these trials require pre-screening where you first have to have your HLA type tested to see if you'd be eligible for these trials.
So what would be an example of an HLA type?
Right, so it's a little more complicated than people know of your blood type, like A positive or O negative, or where it's just a letter and a plus or minus. So the HLA type is a bit more complex, where they will typically test for at least 3 things, and you inherit one from the paternal and maternal side. So for example, HLA A0201. So it's usually a letter and then 4 digits afterwards, or HLA B 0702 or something like that. So this is how the HLA type, it's not really a routine test unless somebody is getting an organ transplant or being an organ donor, or for these types of immune-based studies.
So in the vaccine part, again, okay, this is I'm dumbing it down for me. In this vaccine part, if that's kind of the skin, then the HLA is identifying what is going to graft properly to it, so that we can get in front of the cancer.
Essentially, yes, yeah.
Was that too big of a reach for you?
No, I think it's a good analogy. There are other types of immune-based therapies, which we call cellular therapies, which work a little bit differently. So, for example, there's a company called Iovance that is running a trial where patients who have been on chemotherapy plus immunotherapy get a portion of the cancer removed by surgery. And what we tend to see is when you look at cancer under the microscope, not only do you see the cancer cells, but you see immune cells mixed in with the cancer cells. And that reflects, again, your immune system is trying to fight, or your immune system knows something shouldn't belong there, and the immune system is trying to attack the cancer, but the cancer is stopping the immune system. So these cellular therapy studies involve having a surgery, taking a piece of cancer out of the body. And here what you're interested in are the immune cells. So you separate out the lymphocytes, which are your T cells and your immune cells. You separate them out from the cancer. You expand them in the lab, stimulate them, and then re-infuse them or inject them back into the same patient, with the hope that they already knew how to home in on the cancer because they were already present within the cancer, but now you've sort of outside the body taken the brakes off, you've grown them out into large quantities, stimulated them so that they're really active, and when you infuse them back into the body, the hope is that they'll go and attack the cancer and cause any other sites of disease to shrink down.
And what is that process called?
So those are referred to as TIL therapies, TIL, and it stands for tumor infiltrating lymphocytes. So lymphocytes are your immune cells, like the B and T cells. And so the tumor infiltrating lymphocytes are those T cells that are infiltrating or admixed in with the tumor. So you're trying to take those TILs, expand them outside the body, and reinfuse them to attack the cancer.
Okay, I can see that we are at time, and I want to be respectful of your time. But I also want to shout out to anybody who, if you have a question, then post it real quick, and then I'm gonna. You're being so very kind for taking all this time with us and just doing an amazing job explaining everything to me, to us all. So I really appreciate it. Let's see, I see one question in here. Could you briefly explain the difference between a CT and a PET scan? Because I know
Oh,
it's two
sure.
different things.
Yes, and I just sent a private response back in case we were short on time. But right, so there's a CT scan, and then we call it a PET CT. A CT scan shows you, the anatomy in good detail, and the output, if you've seen your scans on the screen, it's black and white and shades of gray. Sometimes to get a little bit better delineation of, what's a blood vessel, like what's an artery versus a lymph node versus this or that, sometimes in addition to the scan, they administer contrast. And that can be intravenous contrast, through the vein where they inject it before you go under the scanner. And or it can be oral contrast where you drink that liquid that's such a pain, and that kind of coats and highlights the intestines and the bowel. It doesn't get absorbed, but it kind of forms this coating so that you can see and differentiate the intestines from the surrounding structures on a scan. And that gives very good anatomy. And by and large, that CT scans can be used very well for assessing cancer and response to therapy. Sometimes if there are if there's a little bit of kidney dysfunction, or if the creatinine, which measures the kidney function, is elevated, we don't like to administer a lot of that intravenous contrast for the CT scan because it can further irritate the kidneys. It leaves the body through the urine, and so if the kidneys are a bit abnormal, that contrast can accumulate, and it can also further irritate the kidneys. A PET CT scan often is done together. It's a CT, usually without the contrast for CT scans. Plus you overlay a PET. A PET is a different type of an injection. It's what we call a radio-labeled molecule. So it's basically a glowing molecule that is injected, and it tends to accumulate in parts of the body that are more metabolically active, so kind of active parts of the body. So the PET tracer can tend to light up, and here on a PET scan, it tends to show up as a color, maybe depending on the scanner, more of like a yellowish, reddish, orange color, rather than the black and white and gray, or sometimes a little bit bluish. But it goes to normal organs. Your heart is pumping every second, so your heart is very active. So usually the heart muscle glows. The brain, in most of our cases, although sometimes if I'm a little tired, my brain's not as active, but the brain always lights up bright on a PET scan. And this tracer leaves the body through the urine, and so the kidneys and bladder always light up. But cancer can also light up. It soaks up this tracer and shows up as a bright spot. Or infection, inflammation can show up as a bright spot. So a PET scan is very, very good also for assessing cancer. It can take just a little bit of a longer. They don't want you to eat or have any kind of calories or glucose or sugars for at least 6 hours. So it's a little bit of a pain, especially if it's scheduled later in the day. You have to skip breakfast and lunch, and feel hungry for the majority of the day. So it's just a little bit more of logistic planning, but it's also a very excellent way of assessing cancer. So sometimes we use one or the other. The PET can add a little bit more detail to the bones. The bones are bright white on a scan, so sometimes it's hard to see a small spot of cancer in the bone, but a PET can give us good detail in the bone too. So sometimes we can add a little bit more information with the PET scan, but other times the CT scan can give us a perfect view of the bones too. So there's some, again, pros and cons to both scans. Different institutions will use them preferentially differently. So
Okay, great. Well, I don't want to take up your whole evening. I could just listen to you talk all night long, and but you have a real life, and and I so appreciate you taking the time with us tonight to kind of go through and explain all this and with just so patient. It's really, I really thank you so much.
Of course, Terry, thank you so much. I want to extend a sincere and very warm thank you to all of you for also taking your time. You all have lives too that are rich and full, and so taking time to talk
Yeah,
about
well,
this.
we didn't
Thank
choose
you, Dr.
cancer.
Awad.
You chose it. So thank you.
Thank you. Thank you.
So truly, and I'm sure I'm going to see everybody. We're going to see you around, I'm sure, and doing all the amazing things that you do. And so I appreciate you letting me reach out and connect, and you'll be hearing back from me.
Of course, and a huge thank you to all of our patients out there that have been so gracious with your time. Many people have been involved in clinical trials, or allowing us to kind of learn and study and do research, and that is so meaningful to the field as well. So thank you for all of your questions, all of your time.
Thank you so
I
much.
appreciate it. Thank you so much. And I'm going to go ahead and close out the room, and I'm going to send you an email so I can get the mailing address from you and everything. Okay. All right. Thank you all, and I'll be talking to you soon. Okay. Thank you.
Thank you, Terri.
Good night.